I'm Julie Wilder. I'm a scientist at the Loveless Respiratory Research Institute in Albuquerque, New Mexico, and I'm working with Dr. Bobby Scott on a project that is funded from the Department of Energy to study the effects of low-dose radiation on a variety of different things, but our particular project is on the immune system. And so one of the things that we've been doing in my lab with the help of a very talented graduate student whose name is Veronica Bruce. She is looking at the effect of injecting a carcinogen called benzoapyrene into mice, and then after looking in a long-term series of experiments at how that carcinogen induces lung cancer. So we know that low-dose radiation treatment has been effective in inhibiting cancer development in other systems, but not necessarily in the lung. And one of the things we're very interested in benzoapyrene is because it's a major component of cigarette smoke. So cigarette smoke has over 5,000 carcinogens, but benzoapyrene is one of them. So what we've shown most recently is that the carcinogen benzoapyrene predictably induces lung tumors in a mouse strain called AJ mice. And these mice are a little bit more prone than some other mice strains to have lung cancer. So predictably, the benzoapyrene did induce lung cancer in those mice. It takes a period of 46 weeks was the time point at which we looked. Now, if we injected the benzoapyrene on day 0, and then 4 weeks later, we began to treat groups of mice with various levels of low-dose radiation, gamma rays. We would treat them with either no radiation at all, or 10 milligray of radiation, or 100 milligray of radiation, and they were given over a period of six doses every 2 weeks. So, and then that took us to week 14, and then after that, the mice were just left in their cages, and they developed the cancer that they were going to develop. So what we found was that upon euthanasia at 46 weeks, when we looked at the lung cells, the structure, if we looked for tumors in the lungs, what we found was that our pathologist was able to distinguish whether there was a frank carcinoma there, so there was a tumor, a genic event, or preceding that, he could distinguish adenomas or hyperplastic foci. And what we determined was that the actual addition of 100 milligray of radiation, low-dose radiation, actually inhibited the amount of cancerous adenomas that were visible pathologically in the lungs of these mice. And so that was kind of exciting for us because the hypothesis would be that low doses of radiation actually prohibit or can ameliorate cancer cell formation, because low doses of radiation actually stimulate the immune system and also repair DNA damage. So that's kind of cool for us that Veronica was able to show that. Now, as far as the mechanism goes, we're working on that right now. And what she's done in the meantime during this 46-week-long experiment is she has treated mice with BAP, the carcinogen, or radiation, or the combination of the two. And she's looking at changes in the immune system, but in this case at very short-term time points. Rather than waiting 46 weeks, she's waited like 2 days, 7 days, 14 days. And what she's found is that indeed in some cases, the low-dose radiation is stimulating. Actually, what it's doing primarily is that the carcinogen induces an inflammatory response in the immune cells. So we're looking at spleen and lung cells. And what the low-dose radiation does is it sort of dampens the inflammatory response. So what we see is a series of inflammatory cytokines coming up in the spleen cells, for instance, which after treatment with low-dose radiation, that those inflammatory cytokines are either not elevated as much, or the low-dose radiation itself will induce suppressive cytokines. So we're modulating the immune system by kind of dampening the inflammatory response, which might be in the long term a good thing, and B, the reason by which the lung cells were less, there were less tumors in the lung at the 46-week time point. So we have a lot to do as far as getting between week 2 and week 46, trying to figure out all of the different events that might happen along the way in the immune system. But she's working on that. We're also looking into, we don't have any data on this yet, but what we're looking into is the interaction between the BAP and the low-dose radiation as far as DNA damage enzymes go. She's looking into that right now. And we've also have a high-dose radiation. We consider 1 gray or 1,000 milligray as our high-dose control, and that high dose of radiation does have the predicted effects on the immune system that we would think. That is, it's cytotoxic and does not stimulate the immune system. The BAP, actually the carcinogen, we found to be cytotoxic as well, and the low dose radiation can sometimes ameliorate that depending on what type of cells we're looking at in either the spleen or the lung. So we continue with the study, and hopefully by this time next year we'll be able to know more mechanistically about how low-dose radiation actually can inhibit lung tumor formation that's induced by a carcinogen.